Okay, I'm going to start this video back at the beginning of the cartilage section to hopefully let this make more cohesive sense. So we're looking at supportive connecting tissue, specifically with cartilage. Cartilage is a very tough, strong tissue. It serves as a substitute for bone at times. It's smooth, flexible, very low friction. So sometimes where you articulate two moving parts together, it will be used as an in-between surface for those two parts, as we can see here with kind of at the knee or where our ribs and sternum meet. Cartilage evolved before bones, and initially animals with internal skeletons only had cartilage. We see this still with cartilaginous fish like sharks. They just have cartilage and not bone. They're as strong, it's durable, and it's a very major evolutionary advantage when you have to um, support stronger parts of the body. And it was stronger than any other available tissues until bone came along. In fact, cartilage is actually developed first, and then bone is actually coming from cartilage in many cases. And of course, we still have cartilage, but in humans, the bone is necessary for us to withstand this three-dimensional structure that we have with gravity holding on to us and kind of pulling us down. But there are times that we also have cartilage where bone is too stiff. There's only one cell type called the chondrocyte. It's dispersed, unattached. Collagen and elastic fibers are abundant throughout the extracellular matrix. It's a ground substance, in that it's the ground substance around it is very viscous with proteoglycans. Remember, that's just carbohydrates with proteins attached. Now, the cells that are in cartilage are chondrocytes, and their purpose is to secrete the cartilage extracellular matrix that we've just mentioned. So within the cartilage, you'll actually find your chondrocytes sitting in empty spaces called lacunae, or more specifically lacuna. And if you didn't have the chondrocytes in those spaces, they would actually be crushed by the very solid cartilage around them. Now, the protein fibers are the in this collagen extracellular matrix can contribute the most to the collagen strength and hardness. About 2/3 of the mass of the cartilage is protein fibers. Usually, there's only one type of protein fiber in cartilage, but you end up having a lot of that one type. And most of the cartilage in the body uses only collagen fibers. So that is the normal within this kind of matrix. So they're very dense, very tight, which makes them very durable and tough. In one type of cartilage, only found in a few places, you do get some elastic fibers. And in this case, your ear is one of those places. And so it allows for that cartilage to be a little more flexible, a little more bendable than maybe other cartilage. So your outer ear, your epiglottis of the throat are two places where you find this. Okay. Now, unlike your connective tissue proper, there's not much free hyaluronin in the ground substance of cartilage, but there are a lot of proteoglycans that contribute to the stiffness. These proteoglycans that are found in this ground substance of cartilage often have relatively short chains of hyaluronin as the carbohydrate portion. So that is, instead of being free, they are attached. So these proteoglycans are able to bind up water, helping the cartilage stay very flexible but stiff. There are never any blood vessels in cartilage, so all of your nutrients, all the waste that cartilage has, must diffuse. It has to go through the process of diffusion from cell to cell, and it will come from the extracellular matrix to the blood vessel outside the cartilage. So you just don't get blood vessels within the cartilage, but you will in the connective tissue proper that's outside. As a result, the lack of blood vessels in cartilage, chondrocytes actually are very deep and they're very slow to get materials to and from the blood. And because that's the case, the chondrocytes are unconnected to each other. They're in their little chambers. They're surrounded by this very dense extracellular matrix. And um, this combination of factors is one reason why blood vessels aren't going through it, but also it leads to damaged cartilage being very slow to repair. They don't have the materials needed to quickly repair. It's got to be a slow process, if at all. So chondrocytes can't move around. They can't migrate to the site of damage to make new fibers. The blood can't deliver materials. And as a result, you end up with the cartilage being among the slowest tissues to heal. And in many cases, it really doesn't heal if it stays damaged. So cartilage in the body is always covered by a layer of connective tissue called the perichondrium. And we see that on the top layer right above this hyaline cartilage, for example. And it's a layer of irregular, dense connective tissue proper. And the pericardium is not different from any other irregular, dense connective tissue. It's given a separate name because of its location on top of the cartilage. And so it's just location that gives it its name more than a difference in function. The supporting connective tissue is associated with a second type of connective tissue, the irregular dense connective tissue proper. The perichondrium does have blood vessels traveling, so it does get nutrients and pick up waste. There are three types of cartilage that differ in their relative proportions of the chondrocytes, the protein fibers, and the proteoglycan-rich substances. You get hyaline cartilage, which is stiff and flexible, used to reduce friction between bony surfaces. So you'll see this in joints. Elastic cartilage, very flexible. We see it in the ear, for example. And fibrous cartilage, which we haven't mentioned this before, but it resists compression and it's used in the meniscus of your knee and your intervertebral discs. Now let's talk about bone because it's very related to cartilage. And it's known as an osseous tissue. And so osseo refers to bone. As a connective tissue, the bone consists of some cells in an extensive extracellular matrix. There is more extracellular matrix than tissue. Sorry, let me say that again. So there are more extracellular matrix than cells in bone, just like most other connective tissue, which is not surprising. There are four cell types. There are osteocytes, osteoblasts, osteoclasts, and osteoprogenitor cells, basically your stem cells, okay, also called osteogenic cells. The osteocyte is the bone cells that are found in empty spaces called lacunae. Sounds familiar. We see this in the cartilage. That help to prevent the solid extracellular matrix from crushing them. And they have many long extensions that project outward. And so they're surrounded by these little tiny channels, as you can see here, these blue extensions that allows for osteocyte extension outward. And these are called canaliculi. Canaliculi, I always get those words caught up in my mouth. And so you'll see a canaliculus, but notice it says canal. So it's a space moving through these fibers here. You've got the osteocyte bones cells that are connected to one another via their projections. Basically, you get gap junctions connecting and joining them between their cytoplasms. They are involved in the maintenance of existing bone tissue. Unlike other types of connective tissue, they are connected to each other through these extensions. And so even though they are still quite a bit separate from each other, they're still more connected than many of the other cells that we see. Osteoblasts, they help to form new bone and convert to osteocytes eventually. Osteoclasts will dissolve existing bone to allow repair and remodeling. And we'll talk more about how your bones actually do change over time. And osteoprogenitor cells or osteogenic cells are the adult stem cells that can form new osteoblasts and osteocytes, but they don't do osteoclasts. Okay. The bone extracellular matrix has very little fluid and consists of mainly solid calcium, mineral deposits, and collagen protein fibers. The extracellular matrix of bone is pretty cool. It's what gives bone its useful properties. It's flexible, but it's really, really strong and firm. Consists of mineral calcium, mostly in the form of calcium phosphate. It's called hydroxyapatite. Excuse me, hydroxyapatite, and also calcium carbonate, both of which provide strength. Mineral calcium surrounds collagen protein fibers, and that provides the flexibility. Fibers are always about flexibility and connection. The calcium phosphate mineral of bone is very hard, but without the collagen fibers, it would be brittle, kind of like what you see here with the rebar in um, your concrete. And so in this extracellular matrix of bone, it really does act like the rebar, and they make the solid material around them stronger, so less susceptible to shattering, which is great because we put a lot of pressure on these bones. You can see the canaliculi working their way outwards. Here you've got these blood vessels. This is your um, osteocytes and the lacunae, okay, inside these little holes. This is your central canal around everything, which allow the blood vessels through. And then you've got matrix all around this. So just like the outside of cartilage is always covered by the perichondrium, connective tissue, the outside of the bone is covered with a layer of irregular dense connective tissue proper, and it's called the periosteum. In the supporting connective tissue bone, it's always associated with a second connective tissue. It's an irregular dense connective tissue proper. The bone repair is a lot more efficient and speedy than cartilage repair for two reasons. The osteocytes and bone are connected to each other, so you get this connection of cellular cytoplasm, so things can transfer back and forth more easily in terms of nutrients, for example. The chondrocytes and the cartilage are isolated with unconnected like lacunae. Osteocytes can share and transfer materials this way, so it speeds repair, whereas the chondrocytes cannot. Bone repair is much more efficient and speedy than the cartilage repair. So we've got blood vessels travel through the bone, providing access to fresh materials via osteocytes. Basically, you get materials coming into the blood, but you don't get it into the cartilage. Okay, so that makes a big difference in being able to repair bone over repairing cartilage between the bones. Now, when we talk about membranes, this can be a little confusing because in cell biology, we talk about cells. Membranes are a bilayer of hydrophobic lipids and proteins. In tissue biology, histology, membranes are a physical barrier of tissue that line or cover portions of the body. So the term is used inconsistently in biology, but in histology, the term is more functionally defined than structurally defined. So basically, if you can pull a single sheet of it off or cover something with a single sheet, you've basically got a tissue that's considered a membrane. The pericardium around the heart is considered a membrane, but histologically speaking, it's a sac made of connective tissue, a layer on top and an epithelial layer, two different things. But they are tightly attached together, so they come off as one thing. In anatomy and histology, the term membrane is still used more for historical reasons because of how it was used historically. So it's the lining of something, often two layers of tissues, not always, but sometimes you can get one, sometimes you get three in your tissues in your layers. So basically, we call this phenomenological, and that's how it seemed at the time. So that's how it was named. It's not truly histological, where they actually get down and broke apart the different tissues. So histology should always zero in on each specific tissue and distinguish them, even if they are attached. Some examples of the inconsistent use of membranes, serous membranes are two tissue layers, the sacs that surround organs in your thoracic and your abdominal pelvic cavities, for example. So pericardium, pleura, peritoneum. You've got mucous membranes, two tissue layers, and they're the lining in your nostrils, the lips, eyelids, the ears, the genital areas, and the anus. The meninges membrane, which is one layer, and you end up with three separate layers that cover the brain and spinal cord. Each one is considered a single separate membrane. Now let's look at muscles. Muscle tissue is the third broad category of animal tissue. We talked about epithelial tissue, we talked about connective. Muscles are the only tissue able to contract. This is used to create movement, both of the body and the organs within the body. And it's important to realize that muscles can only use force to shorten themselves. They only contract. They can't cause themselves to lengthen. Muscles can pull two things together, but they cannot push. When a muscle stretches, it's relaxed. It's not using force. So when you cause something to contract, you use force. When you relax, you let it go. Relaxing and stretching is a passive, no energy is used, no force is generated, but you get this active contracting, pulling, which requires energy. When something appears to be pushed by a muscle, it's actually being pulled by another on the other side. So to pull the forearm, the biceps brachii muscle contracts. When the forcep is being let down, the bicep brachii muscle relax, but they don't contribute. The tricep brachii has to contract to pull the arm down. So the three broad categories of muscle tissue are going to be defined by function and their cellular characteristics. So you've got skeletal muscles, cardiac muscles, and smooth muscles, and their functions are going to depend on a lot of times by their location. So to distinguish the muscle categories, a few basic cellular characteristics are used to distinguish the three categories of muscles. We've got whether or not they are bands or striated, or so striations, whether or not individual cells with individual nuclei can be distinguished, or whether the cells have become fused, appear as multinucleate fibers rather than individual cells, whether the cells or fiber shapes are long or short or broad or tapered, or also something we're looking for. And you can see some of that down here in these pictures. Here's that fiber shaped, long and tapered, for example. You can see the individual nuclei and connections here. So skeletal muscles, they are responsible for movement. They are generally attached to skeletal components and they're under voluntary control. Okay, you can kind of see the striations in the muscles here. Okay, they're striated. Muscles are multinucleate because each one contains hundreds or more nuclei, usually at the edges. You can see that, especially here. Individual cells cannot be distinguished, instead appear as long fibers that are bundled to create a single muscle. Cardiac muscles, again, only found in the heart. They are also called cardiocytes. They contract continuously and spontaneously. They are under coordinated control of special pacemaker cardiac muscle cells, which start the rest of the cardiac muscles. So we don't control this, it's uncontrolled. So very different shape than what you see in the muscles of your skeleton. They are striated, they have individual nuclei, and they look kind of short and branched. And with the different cardiac muscle fibers separated by a barrier. So you can actually see separations called an intercalated disc. And it's just where two plasma membranes join and you've got little holes through it called gap junctions and desmosomes. So the cytoplasm can move back and forth. Smooth muscles are found surrounding hollow tubes of organs, your digestive tract, respiratory muscle, urinary bladder, reproductive tract, blood vessels. They contract involuntarily, but not continuously like the heart. They are unstriated. They don't look like a bunch of lines side by side, as you've seen in the other cases. They are individual nuclei, and they consist of short tapered cells. So they look very different and much easier to tell apart than the other one, I think. Your nervous tissue is going to be your fourth category of animal tissues. This neural tissue is specialized specifically for conducting electrical impulses from one part of the body to another. And these electrical impulse nervous tissues are not like electricity we use in our houses, which are electrons flowing. In this case, neural tissue electrical impulses consist of ions. You've got sodium, potassium, chloride, for example, moving across your plasma membranes, changing the charge distribution. Okay, so just changing charges are going to be how we create the electrical, if you want to call it, current. So you don't need to know this in detail, just get the concept. When the neural tissue propagates an electrical impulse, the inside of the cell becomes more negative than outside. Positive ions are then going to enter the membrane, making it more positive. And the positive ions will leave the cytoplasm and make it more negative inside. And this will keep going back and forth. So what that does is it creates charge imbalances, which is going to cause movement in this case, which is the movement of ions, which is going to be how we actually use electrical impulses. So we've got two basic categories of cells. We've got the neurons, or the neuroglia, also called the glial cells. Neurons are the individual cells that propagate electrical impulses. Neuroglia act as supporting cells. They repair, remodel neural tissue, providing nutrients to neurons, sometimes interacting with neurons to speed up impulse propagation. Neurons have a very distinct cell architecture. They have a cell body with a nucleus and membrane. They have many little dendrites. You see that down here, these kind of long, stringy things that move away from the main part of the cell. And then you have a long one that's called an axon to help move it to the next, so electrical impulses to the next neuron. Some neurons in the body have axons 1 meter long, making them the longest cells in the body. Most neurons are shorter than that, though. Neuroglia consists of six different cell types that help neurons: satellite cells, Schwann cells, oligodendrocytes, astrocytes, microglia, and ependymal cells. Neural tissue consists of neurons connected in extensively branching chains with axons, one neuron connected to the dendrites of others. And you can kind of really see that here. We get little pieces that stick out to the other side. And the neuroglia will kind of be throughout. For a long time, it was thought that neurons could never be replaced. Once you lost one, it was gone. So when you lost the brain cell, you lost the brain cell. We do know now that there are small pockets of neural stem cells in the brain, and they can replace lost neurons or lost neuroglia cells. So it's not say that you lose them completely, but you sure don't get them back as fast as you might lose them. A single neuron is not a nerve. A nerve is a group of many neurons wrapped in a layer of dense connective tissue proper. The nerve fiber is sometimes used to describe a single neuron, and sometimes used to describe just the axon of a single neuron, the longest part. So that makes it a little confusing sometimes what we're talking about, and we have to try to be clear. Now, this is really interesting. What happens when you have tissue injuries, problems, or damage to these tissues? When you get insult or injury to these tissues, they are going to respond to damage those tissues. As a result, the tissues must respond to restore homeostasis. And so these tissues are going to go through a process called inflammation and regeneration to help restore that homeostasis. Sorry, I had to take a quick break. So let's continue talking about inflammation and then regeneration. Inflammation will get triggered after what we call insults and injuries. So things like blunt physical injury, penetrating physical injuries like abrasions, heat, frost, burns, toxins, infection by pathogens, radiation, and foreign bodies. And you can see that example taking place on the right. Inflammation then occurs as a response to that specific insult or injury. We call it, when it's a specific to this situation, it's called an acute inflammation, where a stimulus causes immune helper cells to do their job, and then you end the stimulus. Things basically get better, you've healed. But inflammation that continues for a long time beyond the need for repair is called chronic inflammation, and you end up with ongoing stimulus. You get cells trying to do their job, but it continues to cause inflammation, and you get this repetitive cycle. And in fact, it's considered an aspect of disease when chronic inflammation can go on, and that can actually cause harm. Now, inflammation has 5 characteristic features, and we've known this for a really long time. So redness, swelling, heat, pain, loss of function. What took longer to figure out was that, there's a cellular and molecular basis that causes these to occur. So inflammation starts when damaged cells start leaking their contents. You get hurt and they start leaking their contents, which then causes lysosomes to break open. They release a degradative enzyme that destroy the injured cell, but also start attacking surrounding tissue. It's a very chaotic situation that's called necrosis, and necro means death. So necrosis is the uncontrolled, premature, unplanned cell death that can occur in response to infection, toxins, chemical injury, and lack of blood supply. The leaking cells release ions and signaling molecules into the surrounding interstitial fluid. And when this happens, that the signaling molecules from this site of injury trigger something called mast cells that are nearby in the connective tissue that release histamine and other signaling molecules. And remember, we talked about histamine. The signaling molecules from those mast cells actually also trigger blood vessels to dilate. Now, remember what happens when you have that epithelial tissue stretching. You release content, and those contents can be mast cells and other like lymphocytes. And so they signal that smooth muscle to relax, allowing the contents of the blood vessels to actually be able to come apart a little bit and allow that stuff to get out, which is going to increase blood flow because blood's going to literally get out of your blood vessels. And so you're going to start to see redness and warming up in those areas. Then the signaling molecules from the mast cells are also going to cause blood vessels in the area to become leaky, as we've been talking about. So more blood to those spaces, and then more blood starting to leak out. Fluid, cells, blood squeezes out between the blood vessels, which is going to cause swelling. And then we're going to start to see immune cells showing up like macrophages and neutrophils. This swelling and signaling actually cause pain, and so your nerves will start to fire. So we get this sensation of pain, and the immune cells deliver to this site by the leaky blood vessels will actually ingest and remove debris and pathogens if they're still there. As the white blood cells ingest debris, they will die. Dead white blood cells and fluid end up becoming pus. In an enclosed space, this would be called an abscess. And then if it's within or under the skin, we call it a pustule or pimple. So that's why all that white stuff is. As debris and pathogens are removed, signaling to the mast cells decreases. So we start to see less response. Mast cells eventually stop releasing their own signaling molecules. The vessels in the area constrict their smooth muscles, reducing the blood flow to the area, and then stop that leaky process, reducing redness, and the inflammation ends. Isn't that a fun process? Now, that cleans up injuries and insults, but the underlying tissue is still damaged and has to be restored. So we have to talk about regeneration, where we have regeneration of tissues, which are going to get repairs and replaced from that injury that are lost. And then we have to start, and also this happens while inflammation is winding down. So we've got to replace tissues that have been messed up, injured, lost, destroyed, damaged. And then as this is winding down with inflammation, we start the process of regeneration. So fibroblasts move into the damaged area from nearby connective tissues, whether or not the damaged area is connective tissue or not. Okay, because remember, the blood vessels are in the connective tissue. Fibroblasts lay down extensive collagen protein fibers as an extracellular matrix. Now, this is more dense and more stiff than your regular collagen fibers, binding it all together. So you may notice that sometimes this is a little bit, it will pull on you when you are hurt and you start to heal because that pulling, they're not as stretchy. The region with this dense, stiff collagen fibers is not like the rest of your surrounding tissue. Eventually it becomes scar tissue or fibrous tissue. And it's a specialized, dense, regular connective tissue. So you can actually see the difference and notice that it pulls more. It doesn't stretch as easy. It's not as pliable. Mesenchymal stem cells, muscle stem cells, and epithelial stem cells will start producing replacement cells for this tissue if they were damaged. And some scars fade as they are replaced by new tissue over time, which can take a bit. Epithelia, skeletal muscles, and connective tissues regenerate well, but not all tissues do. Cardiac does not do well. Neurons do not regenerate. mostly don't regenerate, maybe a little bit. And then you end up with scar tissue remaining. So after a heart attack, for example, you end up with cardiovascular tissue damage that's not fixed. So you get scarring, specialized connective tissue with excess collagen, protein fibers at the site of tissue repair. So internal organs like the heart can also have scar tissue. And in the heart, scar tissue won't contract like cardiac muscles. Remember, if it's tighter, it doesn't move as much, it's not as stretchy. So it actually causes problems in, heart attack patients. So here are your things to know. I'll let you read through that. Please let me know if you have any questions. Okay, here's your characteristics, kind of a diagram to help you remember to um, keep all the connective tissue proper together, the fluid connective tissue and your supporting connective tissues.